All right, for number 1, which feature of Model 1 best illustrates how biological information is encoded on a DNA molecule? So we're really looking at which one best illustrates coding, okay? It is not A. Why is it not A? So you can see the 5' and 3' labels at the end of each strand. However, these don't really indicate, it doesn't show how biological information is coded. These just show directionality. So this shows that DNA is always arranged in an anti-parallel orientation, but that does not indicate coding information. It's not B. All that the labeling of the hydrogen bonds here it shows is just where our complementary base pairs are bonded. But it doesn't show us how they're coded necessarily. C, the lines connecting sugars and phosphate groups. Yes, this is an interesting feature that represents covalent bonds. However, it just shows how our molecules are arranged. The sugar phosphate backbone is arranged. It doesn't show us how information is coded. Rather, the answer here is D. The linear sequence of the base pairs. That's what this is the central role in the coding of biological information. So when we're looking at the actual the linear sequence, okay, this is what tells us the code. No other feature indicated in A through C shows us the feature. So what's really important to know is that the nucleotide sequence or the linear sequence of base pairs gives us the biological information on coding, okay, in nucleic acids, specifically DNA and RNA. Number 2, you guys wanted clarification on number 2. This one, I've kind of broken up the pictures because they don't really fit, but this is figure 1 representing a segment of DNA. Okay, so here's our segment. If radiation can damage the nucleotides in a DNA molecule, sometimes what they'll do is they'll replace the single nucleotide with an undamaged nucleotide, but the question is which of the 4 labeled bonds in figure 1 can be broken to remove and replace cytosine without affecting the biological information coded. Okay, so we're looking at, just a hint from the first one, remember we're looking at the linear sequence. Okay, we don't want to damage this linear sequence. Okay, and our options are W, X, Y, and Z. So how am I going to remove this faulty cytosine without damaging the sequence? If I were to break this bond, which is B, bond W, is that going to help me? No, it's just going to break off of this nucleotide, and now we're going to have a mismatch, okay? So that is not going to that affects the coding of the molecule. That's B. If I were to break X, is that going to affect the coding of the molecule? Yes, because I'm breaking this ability for these complementary pairs to line up. But what's cool about these hydrogen bonds is they can be broken and then reformed without allowing any nucleotide to be removed from a molecule. So this one doesn't really change anything. If I were to break W and Z at the same time, if I were to break W and Z, look at what we're breaking off here. We're breaking off this whole thymine, this whole nucleic acid. And what this is going to do, this is going to induce a whole break in the DNA molecule. And while it will probably affect the DNA information stored here because we're getting rid of this T, opening up an option for folding to occur, for enzymes to come in and attach, this will not help remove our cytosine that we really want to get rid of. However, if I break bonds Y and Z, these 2 covalent bonds, these are going to introduce a break. We can kick out our cytosine here and replace it with a functioning, not damaged cytosine. Okay, so your answer here is going to be C. Number 13, a polypeptide polymer is a polymer of amino acids held together by peptide bonds. Here we're looking at dehydration, synthesis, creating these peptides. The question is, As shown in figure 1, an amino acid must have which of the following properties in order to be incorporated into a polypeptide? Right, and this question is really asking what parts of an amino acid allow it to form a peptide bond, okay? So we're thinking of the properties of this amino acid. We looked at we have two groups on amino acids, all right? We have an amine group and we have a carboxylic acid or carbonyl group. These two groups come together and through a dehydration synthesis, a peptide bond is formed. Which of these properties then should we select? Is it the ability to remain stable in the presence of a water molecule? No, that's not a property of an amino acid. Is the R group compatible with an R group of the last amino acid incorporated? Remember, R groups have nothing to do with sequence of amino acids, so it's not going to be our R group. Is it going to be a central carbon atom that reacts with a nitrogen atom? No, the central carbon atom is just kind of sitting there. What the R group is attached to, it has nothing to do with the peptide bond. So our answer here is going to be the ability to form a covalent bond with both the NH2 group and the COOH group, which is our amine and our carbonyl groups. And this forms a peptide bond, okay? So you can see that the carbonyl, we kicked off an H there, and we kicked off an H. An OH here to form water, and we form this peptide bond, which is the bond between two amino acids. All right, so here we're looking at common features of the illustrated reaction showing the linking of monomers to form macromolecules. This was also known as a dehydration synthesis reaction. Now, which of these features is common showing the linking of monomers to form macromolecules? A lot of students thought that it was B. Two different monomers are joined by a covalent bond, and this is actually not true. While different monomers can be joined by a dehydration synthesis reaction, we aren't seeing, remember we're looking at of the illustrated reactions. These are two different monomers. These are not two different monomers. These are identical monomers. So different monomers are not being shown in these illustrations. However, a difference in monomers is not required for dehydration synthesis. So we can have two identical monomers. We can also have two different monomers, seeing that these two amino acids, 1 and 2, have different R groups. So dehydration synthesis can bring together two identical monomers and two different monomers joined by a covalent bond. However, so that makes number 1, or A, is obviously not correct. Now looking at C and D. D says that monomers are joined by ionic bonds. Now we look at this bond here and this bond here that is formed, and neither of these are ionic bonds. These are both covalent bonds. And when we have a dehydration synthesis reaction, monomers are always joined by covalent bonds. So it is not an ionic bond. These R groups, however, can have characteristics that are ionic, but that does not mean that the bond between these amino acids are ionic. So that makes our answer C. Monomers are joined here by a covalent bond and a water molecule is formed. All right, so for this kind of a question, it's really making you look at the illustrated reaction and not talking about like theoretical knowledge, but as illustrated in this particular reaction, what do you see? All right, number 16, we're looking at 3 different amino acids as part of a polypeptide chain. And then we're seeing so that's before mutation, and now we're looking at after mutation. So the question is, how might this mutation affect the structure and function of the protein? Okay, so we're looking at the primary structure here. This is the sequence of amino acids. However, when we change an amino acid in the primary structure, how does this affect the secondary, tertiary, or potentially quaternary structures? I'm going to start with B. It is not B. True, valine has replaced cysteine, but B says that there will be no change in the 3D folding or function of the protein. We know that when we have a change in the primary structure, this is going to have an effect on the secondary and tertiary structures. And when we're talking about the 3D dimensional folding, this is going to be the tertiary structure. C, since this is a linear section, it does not influence protein folding, therefore there will be no change in protein structure or function. We know that so this is not just a linear section, this is actually indicating what are the amino acids, but because these amino acids have different have variations on their R groups, they're going to fold. So there is going to be a change in protein structure and function. D, looking at D. Now we're looking at the two, let me change my color here. Okay, so in D, we're looking at the R groups. And it says, since the new amino acid is bound on one side by an amino acid with a negatively charged R group here, and by a positively charged R group on the other end, the charges will balance and the protein will fold. This is not true. Why? Because the tertiary structure, or the folding of a protein, is determined by the R groups and the chemical interactions between all of the amino acids in a protein, not just 2 or 3, and they don't balance out. It's all of the protein, or all of the amino acids and their R groups are going to have an effect on the folding. All right, so we can expect there to be an effect on the structure and function of the protein because we're changing the amino acid here. And notice here, this is a sulfur, okay? This SH, this is sulfur, and, or actually it's what we call a sulfhydryl functional group, but it contains a sulfur. And sulfur is unique, and you need to know that this sulfur is going to form a disulfide bridge with another cysteine. Okay, so if there's another SH on another amino acid, okay, you need to know that this sulfur is going to form a very, very, very strong bond here, okay? Now, when this cysteine is replaced by valine, we no longer have the sulfhydryl functional group. So, because the chemical characteristics or the chemical properties have been changed due to the new amino acid, our protein is going to misfold, and it's not going to function properly in the cell. So here is A, and from this question you can infer that you should know, no, you don't have to memorize these different amino acids, but know that the different characteristics of side chains of these R groups. We have sulfhydryl is going to usually form a disulfide bridge, and that's one that you do need to know. All right, next 18, which of the following best describes a structural similarity between these two molecules? And this is RNA at the top, DNA on the bottom. Which best describes a structural similarity? No, it is not A. Okay, why is it not A? Because it says that both molecules are composed of the same 4 nucleotides. If you were to watch my video, which you guys did, you would have found that a structural difference between RNA and DNA is that DNA contains adenine, thymine, guanine, and cytosine. Now, how is this different from RNA? RNA contains adenine, uracil, guanine, and cytosine. So these two nucleotide bases, or nucleotides, are different. So they can't be produced from the same pool of available nucleotides because uracil is inherently different from thymine. Both molecules are composed of the same 5-carbon sugar, which allows each molecule to act as a building block. That is not correct. It is not the same type of 5-carbon sugar. Remember, DNA stands for deoxyribose. And in the name, you hear ribose, deoxyribose. The sugar in DNA is called deoxyribose. And the sugar found in RNA is just ribose. All right, so 2 different 5-carbon sugars. The only difference is that this one has an extra oxygen, and that makes all the difference. So these are not the same 5-carbon sugars. I'm going to skip down to D, which shows you which one is correct. D is incorrect because, yes, molecules contain nitrogen bases and phosphate groups, but they are never used in the synthesis of proteins and lipids. Neither molecule, neither DNA nor RNA is used in the synthesis of proteins and lipids. DNA contains the material, which is then transcribed onto RNA and then is used to synthesize protein, but they're never a monomer in the synthesis of proteins and lipids, okay? So the answer there is C. Both molecules contain nucleotides that form base pairs with other nucleotides. Yes, that is correct. Base pairs, these form base pairs. Allows each molecule to act as a template in the synthesis of other nucleic acids. Because DNA is formed from DNA, so we go from DNA to DNA, DNA from DNA, and then we get RNA from DNA. Okay, so these two, each molecule does act as a template in the synthesis of other nucleic acids. And now getting into this last question, how does variation occur? Someone asked in these first three. I'm also going to talk about proteins for you guys just to make sure everyone is on the same page. Now let's look at, I'm going to first show you the similarities between nucleic acids to first determine what our variation is. Okay, so first remember there's always a phosphate group, a 5-carbon sugar, and a nitrogenous base. That is characteristic of a nucleic acid. Okay, now how can these differ? Variation occurs in two things. One, in the 5-carbon sugar, and two, and then the nitrogenous base. Okay, so for example, in DNA and RNA, DNA has deoxyribose. Deoxyribose. That is the name of the sugar, and RNA just has ribose as its 5-carbon sugar. The only difference there is it's got that OH on it in RNA, and DNA does not have that. Okay, so our difference here is the sugar. Variation can occur in the sugar, which can have different oxygen. It can be methylated. It can have different characteristics of that 5-carbon sugar that give it varying functions. Nucleic acids, or the nucleobase as well, helps provide variation. So an example then, DNA contains A, T, C, G. RNA, options there, A, U, C, G. All right, so these two different characteristics, the pentose sugar and the nucleobase, both allow variation among nucleic acids. Now, what about carbohydrates? How does variation occur in carbohydrates? Now, we know that carbohydrates always have a CHO ratio of 1 to 2 to 1. So while they do have the basic building blocks, there are modifications that can occur on carbohydrate monosaccharides. So the monosaccharides are what make the big differences. So we've looked at a couple of examples. We've looked at glucose. We've looked at galactose. We can look at fructose. These are all different carbohydrates. These are different sugars. The only differences between these are the 3D organization of the atoms around the carbons. So they literally have the same atoms bond together in the same order. But I'm going to draw each of these for you down here. We're going to have glucose first. Okay, glucose. We have an oxygen, and you don't need to know how to draw these. But I'm just going to show you their differences, that they're ever so slight, but it makes an entirely different carbohydrate that actually is broken down very differently in your body. Because sucrose is when we take glucose and fructose and put it together, and you get sucrose, which is that's like your typical table sugar. when we have one monomer of glucose, one monomer of fructose, and you get a polysaccharide, or a disaccharide actually, of sucrose. And glucose and fructose are broken down differently in the body. So here's our glucose. Now I'm going to draw fructose. Now, what's the difference between these? Fructose is a little bit different than glucose and galactose, which I'm going to draw over here next. But it has the same atoms, but they're bonded together in a different order, resulting in different organization of the carbon atoms. And when it is dissolved in water, when it forms this ring, the ring will form a little bit differently. So here you see 1, 2, 3, 4, 5 corners, 1, 2, 3, 4, 5, 6 on glucose. Now when I draw galactose, it is going to look very, very similar to glucose. Okay, so I'm going to actually draw underneath glucose so you can see the similarities. I'm not going to draw a similarity yet. I'm going to show you. Alright, so here we go. This is the one difference between glucose and galactose. That's it. This OH is rotated 180 degrees around this carbon atom, and that makes all the difference. So variation in carbohydrates is due to the attachment of groups at various positions around the carbon. So if we have an acetyl group, a sulfate group, a carboxyl group, we can have our hydroxyl group bonded at different locations. We can have our alkane at different parts around, bonded at different parts around the carbon backbone. But just know that variation in carbohydrates occurs from the side groups, varying side groups attached at different positions along the carbon backbone. All right, now for lipids, I'm going to go to the next page. So I'm going to make a new page here. So variation in lipids. Is containing additional bonds, like double or triple bonds, and this changes the the reactivity of the molecule. So when we look at lipids, they are going to be, if you only have single bonds, so here, if you only have single bonds, it's going to be saturated. Saturated, single is how I remember that. Single bonds here, saturated. As soon as we, and the thing here is that it's saturated with hydrogen, okay? Many hydrogen molecules. When we have a double bond, so let's say we introduce a double bond here in this carbon, it's now unsaturated. It has fewer hydrogens because one hydrogen needs to get kicked off in order to complete the octet rule. This carbon only has room for or only one hydrogen now. All right, and because of this double bond, it's actually going to change the configuration of the fatty acid. So this can be either cis, so let's say if it's unsaturated, we have cis or trans saturation. Cis, in the cis configuration, the two hydrogens associated with the bond are going to be on the same side. So if I'm drawing this, let's redraw this fatty acid tail. Carbon, carbon, carbon. Now I introduce a double bond, okay? My double bond here, oops, I'm gonna draw this a little bit differently. Carbon, carbon, double bond, carbon, back down, carbon, carbon. Okay, so there's my fatty acid tail. What happened here is we only have one hydrogen on these sides, okay? Oops, I can't draw it. So this hydrogen, they're on the same side. So this causes your lipid to kind of have a cis. C for cis, C equals cis. Bend in the, a kink or a bend in the fatty acid. Now what are the consequences of this? It is going to, typically we see cis configuration of fats in normal fats. And actually, this is where you're gonna find like avocados, fats in meats, eggs, dairy, okay? Which eggs and dairy are the same thing. But this is the kind of, this is the configuration of a fatty acid that you're gonna find there. Trans, however, so this is cis, trans does not actually, very, very rarely it occurs naturally. It's created through a process called hydrogenation, which you can think of hydrogenated oils. That's kind of what we're looking at here. Now what happens, what's the difference between a cis and a trans? So let's draw a trans fatty acid, a little bit of, we are gonna see a little bit of back and forth kind of going here on a fatty acid chain. Now I'm gonna draw my double bond carbon. All right, so when I have my hydrogens on a trans configuration, your hydrogens are going to be on opposite sides. These are on opposite sides. Now, why are trans fats usually bad? Okay, and the reason that they're bad is this has a similar characteristic to our normal unsaturated fats. So these can condense they can pack very tightly together, all right, which makes them, when these molecules are not moving very much, they can pack up tightly and they can actually form a solid at room temperature. Okay, so these are characteristics that are desirable. So for example, shortening has these trans fats in them, allowing it to be a solid at room temperature. However, we do see that trans fats have a very negative effect on human health because yes, it's a good way to get a butter-like substance for cheaper prices. However, it's caused, there's a very strong link between trans fats and coronary heart disease. And actually the FDA issued a ban on trans fats in foods. So very, very bad for human health. Yet those are the two types of lipids that we see. Variation comes from the length in the fatty acid tail and the saturation, whether it can be saturated and unsaturated. We have two types of unsaturated fats. We have cis saturated fats, as you can see here. Actually, it's more accurately described here, and trans fats here. All right, and you can infer that based on the variation, which ones are going to be room temperature, or which one are going to be liquid at room temperature? Trans fats. And if this were a single bond carbon, unsaturated fats are going to be a solid at room temperature. Solid at room temperature, solid at room temperature. This is not going to be a solid at room temperature because they can't pack very tightly. So these molecules are going to be a lot more fluid. Even as they're at room temperature, molecules are still moving around, right? And they're not going to solidify at room temperature, whereas these guys are. Okay, so that's our variation. Now in proteins. Variation in proteins comes from the difference in amino acids, all right? All right, so remember we have 20 amino acids. And what makes them different? They have different R groups. So let's look at, we have our central carbon, our NH2, all right, so we have our amine and our carboxyl group, and we are going to have an R group that is also attached on the central carbon. This R group gives us our variation. These R groups can be broken up into several different subgroups, and amino acid side chains can be hydrophobic, hydrophilic, or ionic. Typically your hydrophilic polar R groups, or R groups, are going to be polar, which allows them to be hydrophilic, okay? There are different classifications further of the hydrophilic amino acids, but I'm not gonna have you know these for AP Bio. You'll learn those in organic chemistry someday. We also have hydrophobic. All right, our hydrophobic amino acids are going to be are gonna have hydrocarbon side chains. So for example, they're gonna have a CH3 as their R group. Okay, this hydrocarbon does not want to bond with water, or create any kind of attraction to water. So this is going to be an example of a hydrophobic amino acid, also known as nonpolar. All right, your polar or your hydrophilic side chains are going to be either ionic, so they would have, for example, they might have a hydroxyl group. They might be acidic with a carboxyl group, all right? But know that they have these potentially charged side chains. Also, hydrophilic is going to include, you can have a basic side chain. So let's say you have an NH2. All right, if that's your R group. This is also going to be hydrophilic because they have the ability to interact with a water molecule, making them hydrophilic. And then finally, ionic. There can be hydrophilic amino acid side chains that are also ionic. Knowing if something is ionic, it is charged, either negatively or positively, which allows it to bond ionically with other amino acids. So, positively or negatively charged. Variation among amino acids among proteins comes from the amino acids, causing varieties in the primary, secondary, tertiary, and finally quaternary structure. All right, so two things: amino acids cause variation, and finally are causing variation among the primary, secondary, and tertiary structures. Quaternary structures are the coming together of two subunits, which are two polypeptides. All right. The amino acids can have an effect on this structure, however, only if they're on the outside, not where it interferes. Or where it interacts here, okay? So those are our variations in our 4 biomolecules. Hopefully that was helpful. And if you have other questions, please feel free to reach out. Thanks.